Hello everyone, welcome to today's lecture. In this lecture, we'll learn about the basic concepts necessary for preparing reagents used in microbiological experiments. Here is what we'll cover today. A buffer solution is a solution that maintains a constant pH environment, which is crucial because microorganisms are very sensitive to pH. Thus, making buffer solutions is important. To make buffer solutions, we will first learn the concept of concentration. Finally, we will conclude the lecture by reviewing what to consider when actually preparing reagents based on the concept we learned today. First, a buffer solution is one that resists changes in pH when an acid or base is added due to the common ion effect. Since cells are highly influenced by pH, buffer solutions play an essential role in stabilizing the environment during experiments. For accurate experimental results, buffer solutions are critical. When culturing cells, the cells produce byproducts from metabolic activities that can alter the pH of the solution. However, using a buffer solution minimizes these changes, allowing the cell to grow in a stable environment. In enzyme reactions, enzymes are sensitive to pH changes. Therefore, buffer solutions play a key role in stabilizing the enzyme structure and maintaining enzyme activity. To understand the principle of buffer solutions, let's look at the diagram below. This shows how a buffer solution made of acetic acid and sodium acetate maintains stable pH when acids or bases are added. This is explained by the chemical equilibrium and the common ion effect. When an acid is added, the acetic acid ions from sodium acetate combine with the added hydrogen ion to form acetic acid. This removes the acid's hydrogen ions, preventing a significant change in pH. When a base is added, hydroxide ions react with the hydrogen ions from acetic acid to form water, preventing a change in pH. Next, let me explain concentration. Concentration refers to the amount of solutes present in a solution, which is an important concept when preparing reagents. Let's learn two common methods of expressing concentration used in microbiological experiments. The weight percentage is the amount of solute in grams per 100 milliliters of solution. For example, if a solution contains 5 grams of salt in 100 milliliters, the solution is a 5% salt solution. Concentration refers to the number of moles of solute per liter of solution. 1 mole contains Avogadro's number of particles. For example, if a 1-liter solution contains half moles of sugar molecules, the solution has 0.5 moles of sugar, and the molarity would be 0.5 molarity. Since concentration significantly affects the accuracy of an experiment, it is crucial to calculate it when preparing reagents. Let's summarize today's content by answering a simple quiz about what to consider when preparing reagents before an experiment. How many grams of sodium chloride are needed to prepare a 5% sodium chloride solution with 100 milliliters of distilled water? As we studied earlier, to prepare a 5% solution, 5 grams of sodium chloride is required for 100 milliliters of solution. When preparing buffer solutions, pH adjustments are critical. How should pH be adjusted when preparing a reagent? You can measure pH using a pH meter and adjust it by adding sodium hydroxide to increase the pH or hydrochloric acid to lower it. The molecular weight is essential to know in order to make a reagent at the desired concentration. How can you find the molecular weight of a reagent? You can find this information in reagent catalogs or online databases. With this molecular weight information, you can multiply it by the molar concentration to find the mass of the reagent needed. Lastly, how much TSB powder is needed to prepare 100 milliliters of TSB? Since 30 grams of TSB powder is needed for 1 liter of purified water in the image, 3 grams of TSB powder is needed for 100 milliliters of TSB. Today, we cover the importance of buffer solutions, the concept of concentration, and key considerations to preparing reagents in microbiological experiments. Understanding these principles is essential for ensuring accuracy and reliability in experiments. Thank you for your attention.